Hello, my name is Mak Saito. I'm here to give a talk for Rachel Foster's seminar. Thanks very much for the invitation. Let me start my share screen. There we go. Great. So today I'll be talking about some work we've been doing recently on metalloproteomics and in the Pacific Ocean. And to set the stage, we all know how microbial enzymes are really important in catalyzing biogeochemical reactions. Here's a couple figures from review papers talking about the microbial engines that drive biogeochemical cycles, all these different redox reactions. And over on the right, we have reactions from the nitrogen cycle. Every single one of these reactions is catalyzed by a metalloenzyme, and we'll be focusing on one of these today in particular. One of the big questions I think that we are all interested in is what really controls all of these biogeochemical reactions, and how can we study them and assess the impact of the enzymes that catalyze them directly. And so one thing we've been working on for a long time is developing the ability to measure the enzymes directly using mass spectrometry-based proteomic methods, and that's what I'll be focusing on today. And a lot of this work is inspired by the GeoTraces program, which is an international mapping program for metals and isotopes. This is shown here, the Atlantic Basin and various ocean scale sections going across and measuring dissolved iron in this case. And you can see hydrothermal plumes of iron and very low iron in the Southern Ocean where it's iron limited. And this kind of global scale study is really something that we would love to do for microbial biogeochemistry, and there's a growing effort in the community, a self-organized effort to try and build a program currently titled Biogeoscapes to study ocean metabolism and nutrient cycles on a changing planet. So I encourage you to look out for that and participate in some of the community building efforts that are underway. There'll be a US OCB meeting. It's delayed by COVID, unfortunately, but we'll be trying to figure out when that can be and to get US input. And a variety of other nations are having workshops as well. And as part of this, we're also conducting a metaproteomic intercomparison, and a number of colleagues are involved in that, so that will be ongoing as well. So the method that we're interested in applying is metaproteomics. This is a method that is particularly new and complicated, but we are really excited about its potential. Here's a figure we developed from a best practices meeting with members of the community, where we identified 4 major challenges or major topics of challenges: the sample collection challenge, trying to collect samples from a dilute ocean and getting access to the ocean, and having those samples be containing thousands of organisms per sample. And that's what is the meta of metaproteomics. There's a mixture of organisms present that can't be easily separated typically. And then there's complicated analysis by mass spectrometry and interpretation of the big data sets that come out of it. And then sharing this data with the community is very important as well, in terms of allowing people to access these exciting new data types, and exploring their hypotheses and their research. So previously, our lab had developed a targeted metaproteomic method, as we called it, for biomarkers of iron and stress in marine cyanobacteria, in particular the marine cyanobacterium prochlorococcus. And this was a study we did in the Central Pacific Ocean, known as Metzyme. And in this gray line here is the pigment for divinyl chlorophyll A. This is a pigment that's unique to the Cyanobacterium prochlorococcus. So you can see it's present across this region. But we looked at 2 nutrient stress biomarkers: the nitrogen stress biomarker P2, which is a regulatory control protein, and the iron stress protein IDIA, which is part of the transporter for iron in the cyanobacteria. And what we noticed as we went from north to south across this region is that the nitrogen stress protein was far more abundant in the northern part, but disappeared on the southern stations, and vice versa for the iron profiles. And these, when plotted in sectional format, showed a very consistent pattern with nitrogen stress in the north and iron stress in the south, which was consistent with the distribution of nutrients. In particular, nitrate and phosphorus are known to upwell with equatorial upwelling, and the integrated signal in the upper 100 meters showed how there's enhanced nitrogen on the equator, consistent with the nitrogen stress ending there and the iron stress kicking in. And interestingly, this iron stress continues into the southern hemisphere where we know there's less iron deposition. So this was really the first mass spectrometry-based absolute abundance measurements of proteins in the oceans, and it was successful in determining how these biomarkers for nutrient stress were behaving in this euphotic zone phytoplankton population. And so from there, we wanted to try and examine some of the mesopelagic signals, and I'll be talking about that today. But to set the stage, I wanted to talk about some of the methods that will be being discussed. In particular, we have two metaproteomics data types that I'll discuss today: a global discovery metaproteomics, and this allows us to do deep proteomes. We can do one-dimensional, or more typically, we do two-dimensional chromatography separation. And that allows us to get relative abundances, typically in units of spectral counts. And that's shown here. This is a single sample being injected, and it's basically being separated into across two columns that are orthogonal in their chromatography, and that allows us to get really deep into these samples. It basically looks like 10 or 12 individual HPLC runs as it goes into the second dimension in each case. And this is what this sample looks like when you have all the peaks, and you can see how amazingly complicated it is. We can then, once we finish our discovery mode, we can find interesting targets and switch to our targeted metaproteomics mode, which is used in the example I just showed you before this. And typically now these samples are calibrated with stable isotope labeled peptides, and that allows us to go to units of copies per liter or copies per cell or femtomoles per liter. And that allows us to do stoichiometry as well as make comparisons across space and time, because it's not a relative unit, it's an absolute unit. And just to show you how complicated these spectra are, there's in this one discovery data set, there's roughly 100,000 spectra. So it's definitely a big data type. So for the study I'm talking about today, we combined data from two expeditions, the Metzyme expedition and the Proteomes expedition, that were based on the Kila Moana and the Falcor in 2011 and 2016. And these were crossing the Central Pacific Ocean in regions of low oxygen. You can see the hydrographic data here for Metzyme on the left and for Proteomes on the right. And you can see how we bisect that low oxygen region on this north-south transect of Metzyme. That also overlies an N-star minimum, which is, in this case, the low values are indicative of denitrification. And then there's a little bit of a nitrite maximum there. Interestingly, nitrate is more abundant than oxygen, which one would expect when oxygen is depleted, but you still have you have high macronutrients in the mesopelagic depths. And we also see a strong N15 nitrate signal, which is also a signal of denitrification as well. And these signals, as well as the low oxygen, could largely be ejected from the eastern boundary, but there may be some in situ processes as well, and that's one thing that people are interested in trying to sort out. On the Proteomes cruise, which is an east-west cruise, you can see that we're basically walking up this low oxygen tongue. It's low values all the way across, and the N-star is also relatively low all the way across. And there's some nitrite just below the euphotic zone, and again, the nitrate is far more abundant than oxygen throughout this region. So we collected samples by McLean pumps using in situ sampling methods on the wire. These pumps are able to filter tens to hundreds of liters. In our case, each sample had 100 to 500 liters per sample. These are 142 millimeter filters that we take punches or slices out of, and we basically conducted profiles of them. Today I'll be talking about the smallest size fraction we collected, the 0.2 to 3 micron fraction. And here are the total abundances of protein across 7 profiles from these two expeditions, the Metzyme shown here and the Proteomes shown here. And in each case, we can see abundant proteins in the euphotic zone and lower biomass down in the mesopelagic as is expected. So our global metaproteomics analysis conducted protein identifications by using a custom metagenomic database, and this is through our collaboration with Chris DuPont at the Venture Institute. That metagenomic assembly was collected on, metagenomic samples were collected on the Metzyme cruise, and the assembly resulted in 28 million unique open reading frames. We then mapped our millions of spectra, 100,000 spectra per sample, to these this metagenomic data, looking for matches in mass and predicted mass, and we found roughly over 100,000, 110,000 unique peptides that were identified when mapping to this metagenomic data set at an FDR of 0.1%. And then when we aggregated this onto the protein level, we acquired roughly 60,000 unique proteins at roughly the 1% false discovery rate. And if you consider that we have roughly 60,000 unique proteins that were now identified across about 100 samples, that creates 6 million data points by multiplication, and it's a really nice, exciting, large data set to explore. Here is the biological distribution of taxonomic groups within this sample set as well. And I want to point out that Jackie Saunders is preparing a larger metaproteomic analysis of the global proteome data set from this that will be coming out in the coming year, hopefully. When we looked at the proteins within this large data set, we can look at them by enzyme commission class. So here are the 6 major groups of enzymes. And one thing that really stands out is that in the twilight and deep samples, Klein twilight and deep samples as opposed to the surface samples, we see an increased representation of oxidoreductases. And so these are enzymes that are doing redox chemistry, and was something we wanted to focus in on more. One thing that was immediately obvious to us, that one of these enzymes, the oxidoreductase nitrite oxidoreductase, shown in red in these plots from the proteomes expedition, just completely dominated the metaproteomes in all of our profiles. This is an enzyme that oxidizes nitrite to nitrate. In red are different open reading frames that peptides are mapping to now, so basically proteins that we're mapping to in this instance. These are relative abundance, unnormalized spectral counts, showing just how abundant this protein is. When we drop to the peptide level, we can look at the 2 subunits for this protein. It's also called NXR A and B for the 2 subunits, or alpha and beta. And on the alpha subunit, you can see that we're getting roughly 42% coverage of this protein sequence. Everything in yellow is basically a peptide that's identified that maps to this protein. So we're getting 30 unique peptides and 42% coverage of the sequence. And then for the beta subunit, we're getting 17 unique peptides with 60% coverage of the sequence. So an awful lot of peptides that are mapping just to these 2 subunit proteins. And then when we look at this on the section level, and now looking at exclusive spectral counts where we let each peptide map to only one ORF, we can really see that in this case, the NXR A and B, or alpha and beta, shown here in red and blue, of course, respectively, just really dominate this metaproteome. There's more down here in the noise or amongst the other proteins, but basically, since we're allowing each peptide to only be assigned to one ORF in this exclusive spectral count mode, these 2 are really soaking up most of the peptides, and then peptides that don't map to these 2 sequences now are showing up down here. They're still abundant, but you can see just how completely dominant these are on the relative abundance scale. And then here are the ocean sections of this across that proteome's east-west component, and it's really a tremendously abundant protein in the Mesopelagic. So what's going on here? We looked at the proteome of a culture of a nitrifying bacterium, and we saw something that was very similar. This is the proteome from Nitrospira marina, strain NB295 from the Watson-Waterbury Woods Hole collection. And you can see that the alpha and beta subunits completely dominate the proteome of this culture as well. They're basically some of the top, ranking the top 5 or 6 proteins of this organism. Consistent with, if this organism was abundant enough in the environment, it would also, basically, since the top proteins in this organism are these enzymes, and if those organisms are reasonably abundant, they would dominate the environmental proteome. This is the crystal structure of nitrate nitrate reductase, which is homologous to nitrate oxidoreductase. Um, and people think this is what nitrite oxidoreductase looks like, but we don't have a structure for NXR yet because it hasn't been crystallized. There's some really beautiful microscopy work that was done prior to this, for example, by Speake et al. From Bach's group, and where they observed that there was a two-dimensional structure that they interpreted as being membrane-bound nitrite oxidoreductase that you can see. So these crystals here, and when treated with polyethylene glycol, it formed this nice matrices shown here, and they interpret this as being bound, the NXR particles of this enzyme being bound to the inner side of the cytoplasm and the intracytoplasmic membranes. And so basically this organism appears to be coated in this enzyme. So we wanted to quantitate this further, and so we used our targeted metaproteomic method. This method basically requires calibration, and so the calibration we did involved the high throughput production of N15 labeled peptides, tryptic peptides, and we produce them by overexpressing them in E. coli. The way this works is that basically we have our targeted peptides of interest that we discovered in our global discovery proteome, and then we use those sequences. We created a piece of synthetic DNA, had it synthesized with our peptides coded for in there, and then spacing them with spacers that we know cleave well by enzymatic cleavage. And so they're all in a line here. And then we put a reporter on the end that basically is a known peptide sequence that we can buy standards for as well. And then we overexpress this, and we get roughly 30 peptides per plasmid in E. coli, and then we can start combining our extracted protein and aggregate hundreds of targets into our standard mixture. This appears to be working pretty well for us. It's also a method that's known as Q-concat. In this case, you can see we took 2 batches of this peptide, this protein overexpression, synthetic protein that has our peptide standards in it. And when we digest them, you can see we see really nice reproducibility on a 1-to-1 line, or close to 1-to-1 line, in terms of peak area of peptides. So implying that we're getting, basically similar abundances of peptides that are reproducible from each fraction. The peak areas are different for different peptides because they fly different in the mass spectrometer, but each individual peptide is showing a nice linear response. So that's really convincing that we have good precision in our production of these peptides. And we also have really excellent sensitivity down here. Sorry, this got cut off. This should be in femtomoles sensitivity. So we can then go into our mass spectrometers and measure these peptides. And the way this works is we isolate the peptide mass, what is known as the precursor for the whole peptide. And shown here is 442.2716 plus 2 charged mass. So roughly double that for the single charged peptide mass. And then you have some isotope peaks of this shown down below. And then here's the N15 labeled one. So these were just natural C13 distributions. But then over here we have the entire peptide now grown with E. coli, and it's offset by several mass units. And then we basically use this as our calibration and ratio it to the natural existing light unlabeled peptide, and that is how we get our concentrations. But of course, we don't do this on the precursor level. We actually do it on the fragmentation level, where each one of these peptides is isolated by the mass spectrometer and collision energy is applied, and the amino acids are fragmented into what are known as B and Y ions, where if you go towards the amino terminus, 2 of those amino acids would be the B2 ion, and 3 would be the B3 ion, and so on. And then when towards the carboxy terminus, it's the opposite. The Y2 ion is those 2 amino acids, and the last 3 is the Y3, and so on. And so the measurement is actually a whole bunch of these B and Y ions together, measured, and their signal intensities are summed, and that gives us the quantitation. So there's actually a lot of high quality data and masses that are going into each measurement of each peptide. And all of this is happening very fast and simultaneously with all the other peptides during each injection on the mass spectrometer. And we can see some of the precision of this measurement shown here, where here are the peptides we've been targeting. And you can see that this is 3 depths that they're collected from the environment, and the average and the standard deviation for biological triplicates, basically, or environmental triplicates. And we're getting in almost all cases a relative standard deviation of less than 10%, typically 5% or so, except for one value that was a little bit high for one peptide that didn't perform as well. But in general, we can see we're getting nice precision in this method. And then we can step back and say, well, okay, we've got this measurement of these peptides, but who do these peptides belong to? And if you recall, we were mapping our global discovery metaproteome data to metagenomes from the environment. And that was allowing us to discover the most abundant proteins there. We have developed some software that we call Metatrip. It's on version 2 now and is available on this website. It's actually a working portal you can load your data in, and it runs in real time. And this software lets you determine the least common ancestor for a given peptide. And so in this case, this top peptide here, if it's shaded in blue and has a 1, it basically is found in this genome, or in this case, a single amplified genome. So here are cultured genomes or single amplified genomes, and and you can just basically see all of these different peptides are showing up largely in the nitrospina single amplified genomes, not the nitrospira like the culture isolate I showed before. And that's consistent with what we know. Nitrospira is more of a coastal or sedimentary organism, and it looks like the nitrospina may be the more important oceanic nitrifying bacteria. And the peptides that we were targeting came from the metagenomic data because they were abundant in the samples, and they indeed mapped to this to 3 single amplified genomes for nitrospina. And from this, we can, we're pretty confident that these peptides are giving us some taxonomic exclusivity towards this taxa. So one of the exciting things we can do with this targeted proteomic method is we can start getting estimated biogeochemical reaction rates. And, this is perhaps one of the holy grails of, in chemical oceanography is, can we start getting rates of reactions, right? And in this case, a lot of people have said, well, omics don't seem to be able to get us rates. They're more of, is the gene there and who's there kind of a determination. But we think that we can actually get rates, at least potential reaction rates, from these chemical omics techniques. And in this case, this is simply using biochemistry. We know the protein concentrations. We've calibrated them to be in units of absolute units per femtomole liter. We can calculate, we know the specific activity of enzymes. Those can be determined in the laboratory from protein purifications. We are fortunate in this case that there was some data for nitrate oxidoreductases specific activities. And so if you take that specific activity shown here in nanomoles of nitrate oxidation per milligram of protein per minute, and you can multiply it by the abundance of protein we have in the environment, now converted to nanograms, and you do some unit conversions, you can actually come up with a rate of 85 nanomoles per day of a reaction for this reaction in the oceans based on our actual measurements. A better way to do this, an even better way to do this, is to use kinetic parameters, Michaelis-Menten kinetics, and the substrate concentration. And just to remind ourselves, Michaelis-Menten kinetics give us our reaction rate, and that's equal to our Vmax, our maximal reaction rate of the enzyme, times the substrate, divided by the half saturation constant plus the nitrite, and that creates sort of a Langmuir isotherm shape. And fortunately, again, these constants had been determined for nitrate oxidoreductase purified from marine organisms. And so we're able to calculate our reaction rate, basically inserting this data and using these kinetic parameters and our concentration data. We had to assume a fraction of total protein for that NXR comprised, because some of these parameters were given in total cellular protein. But that said, we are starting to get some reaction rates just based on these kinetic parameters. One caveat that I want to point out, though, is that in this case, now we are doing this in terms of substrate concentration. So the less nitrate is around, the slower the reaction rate. That's assuming first order kinetics, but realistically, this probably should be second order kinetics because these oxidation rates are relative to the substrate concentration and the oxidant concentration too. But we don't have those kinetic parameters currently to do that calculation. Okay, so let's see how this works now that I walk you through how we are doing the calculations. We can use our targeted peptides. We had 7 peptides for NXRA and 5 peptides for NXRB shown here. Each one of these is mapping to a different sequence. They don't have to be identical if there is some biological diversity in the enzyme that's present in the environment, which we know is true based on all those different ORFs that we were mapping to from our metaproteome and the metagenome that was present there. Nevertheless, though, they look to be pretty coherent in vertical structure. And so we can average these peptides in each case, and that gives us our NXRA and NXRB average shown here in femtomoles per liter. And we can then multiply them by the stoichiometry of iron. This is a rather remarkable metalloenzyme with 23 atoms of iron and 1 atom of molybdenum per protein complex. So that stoichiometry is reflected here now on this axis, and that results in roughly, 1 to 3 picomoles of this enzyme present in the environment has in the particulate phase of the microbial population. Then we can take these concentrations of enzymes and use our Michaelis-Menten kinetics and combine them with the nitrite concentration in the environment, and that gives us an estimated NXR activity shown in this plot here. And just pointing out that this is actually in a region of relatively low oxygen, but not zero oxygen in this part of the Central Pacific Ocean. So this is at station 3. This is the station that had the most NXR. We did see some variability between profiles, and we believe that is shown here. Here's our all of our stations for the 2 expeditions. In general, the Metzyme stations had higher abundance than the proteome stations, and we think this may be related to a very strong El Nino event that was happening during the proteomes expedition, and so maybe there was warmer waters and more thermal stratification and less export and less enzyme as a result. But one thing you can see clearly is that the 2 subunits are really coherent across this, as one would expect, since they have a 1-to-1 stoichiometry and the organisms are making them. There's some fine structure that varies by different stations. It's surprisingly variable by station, and our interpretation of that is that this is probably a pretty dynamic mesopelagic ecosystem that's responding to various export fluxes, and that allows the population to grow in response to that. We can then also use the nitrate concentration, and that gives us reaction rates across this region, and you can see they vary roughly a few tenths of nanomoles per liter per day. Okay, so what's going on? How do we interpret these reaction rates? And so this table here summarizes these calculations, and we're calling these potential reaction rates because we have to make some assumptions about the substrate concentration. Is it replete or is it limiting? And the different values we use can bracket the range of rates. And so if you remember, that first number I calculated with the specific activity is really assuming a saturated nitrate concentration, and that gives us this maximal rate of 85 nanomoles per day. So that's on the top end of what's possible for this much enzyme in the environment, assuming no oxygen effects, and assuming nitrate is really abundant. But we know nitrate's not abundant, so we can do a couple approaches. One, let's say, what's the maximum nitrate we observed there, and it was 0.5 micromolar at that station 3, and that gives us 5 to 6 nanomoles of oxidation per day, and so maybe that's sort of at the high end of what's realistically happening, at least at a steady state basis. But if we use the actual in situ nitrate concentrations, from these profiles here where the peaks are, there's actually, this is not where the maximum nitrate is. It's usually in a different spot, more shallow, and that gives us rates that are in these tenths of nanomoles per day, right? And then we can compare these to isotopic-based nitrification rates that Alison Santoro conducted on the same Metzyme cruise expedition, and these are ammonia oxidation rates, and if we assume that that goes to completion in nitrification, that ammonia is converted to nitrate, and the nitrate is then oxidized to nitrate with this enzyme, that yields 5 to 34 nanomoles per day at these stations 1, 3, and 5. And so that's sort of in the middle of this range here, right? And so what's going on here? I think we can, based on the cellular proteomics and the prior microscopy and these values, I think we can interpret this as there being an excess capacity of nitrite oxidation enzymes in the Mesopelagic Ocean that are basically waiting for export events. These cells are covering their membranes with this enzyme, and when there's an export event and organic matter sinks and that organism is remineralized and ammonia is released and nitrate becomes available, they're there and ready. They don't need to synthesize the enzyme. They're there waiting for it, and when that comes in, they're able to immediately capitalize on it and react. And so, the steady state concentration, when there's, at the ambient nitrate may be quite low, but if there's a flux, a pulse event that comes in, they're ready and they can ramp up to 85 nanomoles per day, a couple orders of magnitude higher pretty much instantly, because the enzyme is there waiting to generate energy from that incoming nitrite. So another observation we had was that these subunits, this enzyme is more abundant when oxygen is low, and that can be seen here, where we have more NXR when oxygen becomes scarce for both subunits, with the exception of some stations in the southern hemisphere, this station 6, which was shallow. But in general, we see this inverse trend, and it's not because there's more nitrite in those regions. The nitrite is depleted, as I showed in that profile before. So we have a couple interpretations for this. One is that there's more NXR where there's more biomass export and there's more regeneration and then more nitrite, but that's also basically why the oxygen is low. And then a second interpretation is that we know that the NXR enzyme is full of iron sulfur clusters, here 5 iron sulfur clusters, and these iron sulfur clusters are known to be very susceptible to oxidation by oxygen, and so this enzyme is more stable when at low oxygen levels. And these two explanations are not mutually incompatible. They both could be occurring simultaneously. This enzyme probably persists because the oxygen is low, and the enzyme's probably there because there's more export and the oxygen is being depleted. So to step back and talk about some of the interpretations and implications, this is an enzyme that's incredibly abundant in the Mesopelagic. It's more than 60 billion copies per liter. It looks like it's pretty dynamic in its changing abundance, and that implies that there's an influence on surface export and perhaps even climatic processes like El Nino. For us, this was a really big surprise. We didn't expect to see, one of the most abundant, if not the most abundant protein in the Mesopelagic of these oxyminimum zones being a metalloenzyme with 23 atoms of iron, given how scarce iron is in the oceans. So that really, that was sort of humbling for us to realize how little we knew. I mean, certainly there have been reports for transcripts and genes that had NXR in them, but to see this on the protein level was really a surprise for us. It's also interesting connecting this to the topic of N2O, and this is more speculative, but I think it's worth bringing up. N2O is present throughout the Central Pacific oxygen minimum zone, and that's shown here in this profile of N2O here in black. Incidentally, cobalt has almost the identical profile, perhaps due to remineralization structure, or perhaps due to some sort of interaction. We know that vitamin B12, which contains cobalt, is a really strong binder of N2O. N2O is produced by both the ammonia oxidizing archaea and the nitrate oxidizing bacteria, those nitrifiers, albeit through unknown mechanisms, but that raises the potential for, if deoxygenation increases, would that increase the nitrifier distribution and affect the N2O distribution as well? And if that's true, are there also potential unintended consequences if people start raining iron from above in ocean iron fertilization efforts, basically dropping iron into this mesopelagic that has a has an iron demand which may increase with deoxygenation and may interact with N2O in ways we don't really understand. So there's a lot of implications there. I think a lot more questions than answers at this point, but certainly changing the way that I thought about this environment. And from an iron perspective, the iron biogeochemical cycle is thought to be, we've focused largely on the euphotic zone and dust inputs and things like that, but there's a continuum between particulate and soluble and organic and inorganic, and there's a potential that these really abundant metalloenzymes may be an important reservoir, and we can do that calculation. This NXR is 1.3% of the total protein in this area. It's about 1 to 2% of the particulate iron present in this region, but in general, 80 to 100% of the particulate iron is thought to be, or known to be, lithogenic, basically dust type particles that are sinking through the water column, and as a result, largely biologically unavailable. The dissolution rates are quite slow as it's sinking through the water column. So these metalloenzymes may be an important biogenic particulate iron phase in the environment. And so with that, I'll wrap up, but I just wanted to also point out that we have this new website that is intended to help researchers and students and educators explore this exciting new data type of proteins in the environment. It's called the Ocean Protein Portal. And basically it's a way for people to come in and examine where a protein is in the environment. And you can type your protein in here or enter a sequence or something like that and explore metaproteomic data yourself. And so happy to answer more questions about that, but you can find the website at oceanproteinportal.org, and we have a couple papers coming out about it in the Journal of Proteome Research that discuss it. And in particular, I want to make a plug for anyone who's doing doing education, be it graduate classes or undergrad classes or even high school classes. And if you want to explore using this portal as a means to learn about biochemistry or microbial ecology in the oceans, we'd love to collaborate or help you do that, or answer any questions that may be related to this. Okay, so with that, I'll want to acknowledge people. In particular, I want to acknowledge Matt McKelvin and Don Moran for their two technicians in the lab who analyzed all of these metaproteomic samples, and Jackie Saunders as well for some of the data analysis shown here. And then our collaborators, Chris Dupont and Alison Santoro in particular, for the MetZyme projects. And with that, I'll be excited to hear the question and answer session.